Hi everyone, this is Ramalinga Prasad Kuppa. Welcome to my channel Pharma World. Today's topic is ICH Q1 Part 1, Consolidation of Existing Guidelines ICH Q1A to F. The information from ICH Q1A to Q1F and Q5C guidelines is consolidated into one single guideline with an elaborate explanation for each section. In this part 1 video, basic principles of stability requirements are explained. Since the guidelines are too elaborate, the entire guideline will be divided into several smaller video programs. This video is limited to up to section 7 of the draft guideline of ICH Q1. Let us look into the salient features of the new ICH Q1 guide. A requirement of science and risk-based approach is included in this revised guideline. In the new revised guideline, there is a reference to risk-based approach in addition to a scientific base. There are 9 tools in ICH Q9 quality risk management principles on which the strategy should be developed. Use of flowcharts, check sheets, process mapping, cause and effect analysis, etc. should be used for establishing the test conditions. So the stability study protocol should refer to the risk assessment report that would guide on the specifications to monitor, packaging scheme, etc. The risk assessment should consider product knowledge, which is useful in the design of formal stability study protocols. Development studies may also be considered to characterize the physical, chemical, and biological changes likely to occur with storage to establish the degradation profile and intrinsic stability of the product to confirm and validate the stability indicating nature of the analytical procedures. The QBD principles as described in ICH Q8 to Q11 and Q14 are also a part of requirement to the design of overall stability strategy. Quality by design concepts are being used prominently in the process validation activities. Same design space should be considered as a successful strategy for stability studies also. The guideline ICH Q8 to Q11 and Q14 would greatly support a comprehensive approach for stability studies. If the stability information is generated under a more severe climatic zone storage condition, it would be acceptable in the other climatic zones. This is a comfortable point. As we know, there are 4 different climatic zones classified as Zone 1, Zone 2, Zone 3, Zone 4A and Zone 4B, etc. Among all the conditions, if a worst case is selected with higher temperature and higher humidity conditions, the data may be accepted for other zones too. The recommendations may be applicable to drug substance intermediates and drug product intermediates. Drug substance intermediates and drug product intermediates are also a part of this new requirement. A broad spectrum strategy for stability studies include supportive studies, the primary stability studies, commitment stability studies, ongoing annual stability studies, product life cycle stability studies, example to support the PACs, that is post approval changes. So the stability study is not a once-off activity. It refers to the life cycle management of drug substances and drug products. It encompasses post-approval changes too. This is the formal stability studies pattern. The supportive stability studies include photostability studies, in-use short-term storage condition studies, etc. The primary batches selected for study should be similar or representative but not necessarily identical to the process of a production batch. The manufacturing process for the primary batches of drug substances may be similar to the commercial route of synthesis. That means the chemistry should be same, but scientifically justified changes in extraction solvents, additional physical steps, etc. are acceptable. For supportive studies, chemically synthesized drug substances should be manufactured by the same synthetic route. Changes to the manufacturing process parameters should be scientifically justified. Drug products should be of the same formulation and method of manufacture. Batches of drug product included in stability testing should be derived from different batches of drug substance to account for variability in drug substance batches. This is important. This helps to establish study on potential variability from one batch to the other on the quality of the drug product. The strategy may be extended to usage of drug substances from other suppliers also. When the product, process and production of two or more sites are comparable, the retest period and our shelf life would not need to be re-established at an additional production site. This is important. If the data from each site is comparable statistically, there is no necessity to carry out the stability studies separately for each site. A detailed risk assessment should be done to determine the validity of the data from each manufacturing site. For continuous manufacturing process, the batches may be selected from a single startup shutdown sequence provided the statistical variability is taken into consideration. There are some manufacturing sites who make continuous manufacturing. The batch is decided on specific start time and end time. Say every shift of 8 hours or 12 hours or a day of 24 hours. The statistical data and risk assessment should decide this. Let us look into the container closure system. The adverse interactions, if any, between the primary packaging and the drug substance or drug product should be established. This point was referred as a generic requirement in the earlier ISH Q1A R2 guideline. In this, the requirements are elaborated and it should be part of stability study to establish that the primary packaging and in some cases secondary packaging too is suitable for the drug substance or drug product. Impact of packaging component from which the matter may migrate into the product. Example, ink or adhesive from labels should be considered. The evaluation should extend up to the ink that was used for label printing and the adhesive used to affix the label. Changes, if any, in the packaging system should also, during that means the product life cycle stages, be considered for the ongoing stability studies. In case the packaging scheme is revised, the ongoing stability study should include evaluation of the new packaging scheme also. Let us see the testing frequency requirement. The testing frequency should be as prescribed below. 1. Shelf life 12 months or less. The testing should be monthly for initial 3 months and every 3 months thereafter. This is revised. The testing frequency for products with a shelf life less than 12 months, this is the requirement. Shelf life greater than 12 months is every 3 months for the first year, every 6 months for the second year, and annually thereafter. This second point is same as the earlier requirement. Sterility testing or alternatives, example container closure integrity testing, should be performed at a minimum annually, including initial and at the end of the proposed retest period or shelf life. This is an annual monitoring requirement. There are no significant changes in accelerated test study conditions. The test conditions for accelerated and intermediate conditions are the same as in the earlier guideline ICH Q1A-R2. Information of ICH Q1D, the reduced testing is also same. No significant changes. However, the concept is explained well. The brief information in ICH Q1D is explained in an elaborate way here. Annex 1 details the requirements of the reduced stability study protocol. Section 15 prescribes for product life cycle stability studies. The general principles referred in this guide are for the dosage forms. However, the reduction strategy for drug substances may also be explored with scientific and statistical fit data. The information from ICH Q1A to F is entirely extracted into this revised guideline. Since this is in public consultation stage, it is recommended that the stability studies groups to go through the guide in the entirety and comment on any additional requirements or modifications to the regulatory agency to get the best out of this. As explained, the revised Q1 is very elaborate in almost 18 sections. Let us understand the content of some more sections of the guideline in the next part of the video program in the next available opportunity. Thanks for watching. For more videos, please do subscribe, like and share. Also, please leave a message in the comments box for any further support. Thank you.